Hello, everyone. I'm Joanne Bath. I'm the CEO of the Dalhousie Medical Research Foundation. This is our third annual Impact Reception. Of course, this year we're doing things a little different. In the past, those of you who would have attended would know that we would share some food, some hugs, some laughs, and it would be a little less formal than we are here today. But our Impact Reception is an opportunity for us to report back to you, our community, on the impact that philanthropy is having on health research. So thank you for joining us today. Of course, we had to move to an online format this year, given the impacts of COVID-19. Of course, we want to ensure that your safety, our safety, the safety of our researchers and donors is at the forefront. However, today will be interactive. We ask that you post comments and questions throughout the event via YouTube and Facebook. There's comment sections there. We'll be monitoring those, and to the best of our ability, we'll answer those questions either today or after the event. We'll attempt to answer them, and also we'll have some prize giveaways at the end, or at the middle actually of our agenda. So feel free to ask questions, especially when our researchers are presenting. First of all, I'd like to say thank you to classical guitarist Nevin Prostren. We really love that, and I see some of the comments that I think the audience enjoyed your performance as well. And we really appreciate you taking the time to spend with us this afternoon. Nevin's partner, Brianne Cameron, is a PhD student, actually, in Dr. Alex Quinn's lab. Alex is a cardiovascular researcher, and we're so pleased that Brianne works in his lab, and we're glad to have Nevin here today. Now more than ever, we recognize the importance of health research and its power to advance health outcomes for everyone. We know that COVID-19 took many people by surprise, and created a worldwide pandemic that changed the face of how we view health and life in general. And certainly, as I said, how we value research. The DMRF team did not stop working for one day of the pandemic. While we're all at home, we're all still working very hard to raise important funds for medical and health research. And many of our researchers continued their work throughout this. And in fact, many researchers returned to their physical lab just this week. Those working on COVID-19 have been in their lab the entire time. Nimbly, DMRF was able to mobilize $250,000 from our endowment to support COVID-19 research, which unlocked another $600,000 from Research Nova Scotia. Also, DMRF was a leading partner in the creation of the COVID-19 Health Research Coalition, a powerful fundraising consortium that brought together 8 research partners to pool funds to support crucial COVID-19 research. Together, we assembled $1.5 million in funding, and were able to support 40 outstanding research projects, which you can find out more about on our website. We've received word that things are moving forward on the vaccine front at Dalhousie. I know many of you are wondering how that's going. In particular, Doctors Alison Kelvin and Roy Duncan are collaborating with industry on a DNA vaccine. Early results look promising, and they've leveraged their coalition grant into a $4.2 million CIHR grant in order to move forward to phase 1 and 2 clinical trials in the next few months. None of this would be possible without you, our donors. And we want to thank you today for your support. And we want to celebrate the impact that you've had, not only on COVID-19 research, but on so many other local health research projects. This year alone, DMRF granted $5.6 million to critical health research projects at Dalhousie, which represents our greatest granting year yet. So thank you so much for supporting that. From studentships, equipment purchases, visiting lectures, and groundbreaking longitudinal studies, your giving is advancing health in our region and beyond. As you may know, DMRF supports progressive health research, research that spans discovery science, clinical science, social sciences, and health system improvement. We understand the health outcomes of tomorrow depend on the research excellence of today. Your donations to DMRF influence the diagnosis, treatments, and cures of the future, and have influenced many of the discoveries that help us all today. This afternoon, you'll hear from several of our researchers that you've helped to support by being a DMRF donor. Our latest impact report details stories of your giving. You can read this online and view its accompanying videos by visiting dmrf.ca/impact. Beautiful hard copies have also been sent in the mail. I know some of you have received them, but with postal delays, you may not have quite gotten it yet, but it may be sitting in your mailbox this afternoon, so have a check when we're done today. Because of the global pandemic, both versions of the report were done virtually with telephone and email interviews, as well as videos created through online mediums like Zoom and Microsoft Teams. So I just want to say a special thank you to all of the Impact Report participants who virtually invited us into their homes or offices to share their stories and research impacts with us. For those of you who attended last year's Impact reception, you will know that we put our researchers to the test by having them present using what we call the Ignite Talk format. Ignite Talks are a series of speedy presentations where presenters get 20 slides, which automatically advance every 15 seconds. The result is a fast and fun presentation, which lasts 5 minutes. Now, we realize that this is a lot to expect from what is often considered heavy and complicated content, but we're pretty confident these researchers can pull it off. Feel free again to ask questions during their presentations by commenting on Facebook, YouTube. We'll try to answer some of those about the 3 Ignite Talks as we go on. And again, you'll be entered in to win some of our prizes. First of all, let me introduce Dr. Thomas Pulinilkunnil. He's a cardiovascular researcher, an associate professor at Dalhousie University, and an adjunct professor at the University of New Brunswick. Thomas was a research participant in DMRF's 2019 Spring Molly Appeal, which supported the Dalhousie Medicine New Brunswick's BioBank, which is a shared facility between researchers in New Brunswick and in Nova Scotia. The BioBank stores heart tissue samples from patients across the Maritimes, allowing researchers to explore what contributes to heart failure, why some are more susceptible to heart failure than others, and the potential to develop new therapies. Welcome, Thomas. The clock starts now.
Thank you, Joanne, and thank you all of you who are viewing in right now, and a big thanks to all the DMRF donors who has made this event possible and allowed researchers like us to present a glimpse of our research to you. Could I view the slide, please? So that's where I am located in St. John, New Brunswick. That is Dalhousie Medicine, New Brunswick building. I am, as mentioned by Joanne, I am an associate professor in the Department of Biochemistry, Dalhousie University, and I also have an adjunct professorship status in University of New Brunswick. I have no conflict of interest or for this program or presentation. I'm a collaborator and consultant for protokinetics a screening program. As far as heart disease goes, there are 10 key risk factors for heart disease involving diet, genetics. There is also coronary artery disease. There is hypertension. So amongst this, I'll focus on obesity and diabetes. The reason being, once upon a time, our food used to look like this. That is pretty lush green, colorful. However, in the last recent years, there has been a big transformation in how the food looks like, and it now looks like this. So we have food wrapped in wrappers, and it is mostly processed, which has added to the epidemic of obesity and diabetes. So not only has the food composition changed, but also there is a lack of physical activity, and also there is increase in portion sizes. As compared to 20 years ago, you can look at how burgers, fries, and pop they are much more in volume and content, and at the same time we have a phenomenal a lack of physical activity. We are pretty much sedentary in our daily life, adding to the problem of obesity and diabetes. And so the question is that what does obesity and diabetes bring into the picture, and what does it cause? It causes many comorbidities, and one of the biggest comorbidities is cardiovascular disease, which is the focus of the research for our group here, and our colleagues in Halifax. In fact, it is now referred to as the epidemic of the century, and we never thought COVID would beat that. But anyways, to give you a few stats here, its numbers are tripled. It is the risk factor for type 2 diabetes, and in fact, you can see that with obesity and diabetes, the exponential risk of having cardiovascular disease increases by almost 4 to 5 fold with increasing number of risk factors such as smoking, heart failure, smoking, alcohol, and also hypertension. And so diabetes augments the risk of dying from heart disease. And if you were to combine the death of having cardiovascular disease across Canada, and the cost and the economic burden, it would be around $190 billion. And if you were to segregate them, it would be 22 million for diabetic patients. Who are the most vulnerable? Well, they are the pregnant, they are the ones who are children, and also the elderly. They are the most vulnerable patients who suffer from obese and diabetes related comorbidities. And the major reason for mortality in these patients is hypertension and heart disease. And in fact, there are many other macro and microvascular complications, but heart disease is the number one reason for mortality in obese and diabetic subjects. So the question is then, how do we tackle this problem, and how do our group, and what are the different types of comorbidities that we experience? And as I mentioned, there is respiratory dysfunction, and also there are issues at the level of the heart that we try to handle at IMPART and CVRG. And our mission is to accelerate and advance discoveries, explore mechanisms, translate effective therapies, and these are the folks who actually make it happen. This is our team that actually make diagnostics and risk characterization. They discover new mechanisms and they translate their discoveries into the patient population. And these are researchers, staff, and trainees from Dalhousie University. And to just name a few who were part of the Molly Appeal 2019, we have doctors here, Ansar Hassan and Dr. Lachmudial, who look at operative outcomes. We have arrhythmic heart disease specialists. We have individuals who look at different types of cardiac complications. So what's the biobank? What's the tissue biobank doing? Tissue biobank is a process by which when patients are admitted to the hospital for elective cardiac surgery, their tissues are being collected and stored in a freezer, and then this tissue is made available to different researchers. We have 1,000 square foot translational biomedical lab at St. John Regional Hospital, where we store these tissues. And now we as a group, the Impart CVRG group, we look at these tissues, we examine them, and we find cause for heart disease in the lab. And these are this is just a image of where the tissues are stored in the St. John Regional Hospital. What you see are the freezers, and we are very pleased to announce that with DMRF donor support, we were able to hire our first biobanker, Dr. Christy Aguiar. And not only have we established the biobank, we have also started publishing and disseminating our results to the scientific community. And these are some of the publications and some of the knowledge advancements that we have made in the last 3 to 4 years with our dedicated staff. So the impact of this biobank is that it can have broad reaches in maritime community, both in terms of health and research, innovation, discovery, commercialization, and also it can be felt across all this spectrum and can provide a solid backing for helping device treatments for cardiovascular disease. So we once again thank the DMRF donors and the DMRF for allowing us to create this biobank, and also to ensure that its sustenance, its success and feasibility remains over time. And I would like to acknowledge our team who have been very supportive of this endeavor, and their hard work has helped us to make this happen. So once again, thank you for your time, and thank you for your attention. I'd be very happy to take any questions.
Hey, Thomas, thank you so much. I know that that is rapid fire and that it's a lot of information to get out. With my Newfie accent, I think I could beat the 5-minute mark, but that I know it's tough. And you gave us a lot of really important information there. I do have a question for you.
Okay.
Why are diabetics and individuals with obesity issues at higher risk for developing heart disease?
So usually whenever you have obesity or diabetes, it's a kind of an endocrine disturbance. You have your set point in your body for metabolizing certain nutrients have changed, and then as a result, if those nutrients are not metabolized in a proper manner, they flood around in your system. They create waste products in your cells. They do not allow your organs to function well. Your blood flow is compromised. So there is a conglomerate of issues that happen in an obese and diabetic environment that primes the heart for having eventual dysfunction and failure. So and this mechanism is multifactorial. There is not a single point mechanism.
Thank you. How will Dalhousie Medicine New Brunswick's biobank help in mitigating cardiovascular disease problems in diabetics and those with obesity issues?
So one of the things is that we always talk about bench to bedside, the way the therapy works. But what we would like to do is the reverse, bed to bench. And so basically what we want to do is take the tissues from the patients when they are having that particular disorder and have the opportunity to study what's going on in the tissue at the time when they are affected. And if we are able to piece together that puzzle, we are confident that we will be able to devise strategies and have a better understanding of by having a better understanding of the disease. And we have a team of researchers with us who have specialists in certain aspects of the disease, and then we can all come together, and that could help us devise new strategies for treating cardiovascular disease.
Great. Thank you so much, Thomas, for this important work that you do. It's fantastic to be able to have someone from our Dalhousie Medicine New Brunswick campus join us for an event. And we really appreciate you giving us a taste of your research. And of course, you can learn more about Thomas's work on our website. To our viewers, we want to hear from you. So please do keep asking questions. We have a comment here from someone that says, we're very lucky to have you working on cardiovascular research at DMNB. And we are. We also have, do we have any other questions from the audience? Let me check. I don't think we do. So with that, Thomas, thank you so much.
Thank you for having me here. Thanks a lot.
Fantastic. We do have one comment here that I just wanted to pull out. John Archibald, who's one of our researchers, says, a big thank you to the amazing team at DMRF for all that they do for biomedical research. It's true that they haven't skipped a beat during the COVID-19 pandemic. Much appreciated for your efforts. And John, we exist to fund research. And like I said, at this time more than ever, we recognize the importance, and we are so driven and passionate about this work, and we just want to do more, and we know that our donors want to do more. So thank you for that comment. Now I'd like to introduce Dr. Paola Marcato. Dr. Marcato is an associate professor in the departments of pathology and microbiology and immunology at Dalhousie University. She is a recipient of DMRF's Breakthrough Breakfast Mid-Career Accelerator Award. Dr. Marcato is using this award to perform genome-wide assays on cells and patient samples to identify important factors in breast cancer progression and treatment response. Welcome, Paula. The clock starts now.
Thank you, Joanne. It's a real pleasure to be here. I so much appreciate the donors who made this research possible. And so today I'm going to tell a little bit about what we've been doing with those funds. So as soon as the presentation starts, I'll be happy to do my 5-minute sprint. Okay, so today's talk is titled "The Missing Link," and that's not a typo there. You'll see in a minute. Identifying novel treatment targets for breast cancer. And so I'm a breast cancer researcher, and that's what I've been doing for quite a few years now. So breast cancer unfortunately affects 1 in 8 Canadian women. And despite pretty good treatments, a quarter of those women will end up passing away. So that will mean that in Canada, 1 in 3 Canadian women will die of breast cancer. So we need to do better. So my research lab, and I'm showing you here a couple pictures of my research, my students doing research. We're going to try to identify genes which cause breast cancer to grow and spread, so that we can then identify novel drug targets which may lead to better treatment. So our genetic code, our genes define us and are a major factor in disease, including cancer. So when you have a disease, it's typically caused by our own genetic makeup, our own genes, and our environmental exposures. So in the 1990s, the Human Genome Project was started with the goal of sequencing the entire genome. That was a massive project, 13 years, and it cost $3 billion, but it was completed. And it was the hope that it would lead to a revolution in diagnosis, prevention, and treatment of most, if not all, human diseases. And so we're getting there. But I'd like to say that what we learned when we did that sequencing was that actually we don't know a whole lot about much of the genome. Much of it, we had no idea what it was doing. That's what we learned. And people thought maybe it's just junk DNA. Maybe there's nothing there. Maybe there's just regions that are functional and the rest is unfunctional, just spacers. But what we're learning, in fact, is that it's not junk. We just have to figure out what it does. Next slide, please. So what we did when we looked at the parts that we didn't know what they were doing is that there were 12,000 long non-coding RNAs as part of it. And these link RNAs are maybe doing something. And what are they doing in the context of tumors? Well, we studied one of them, which we named NRAD1. And what we learned is that when you target NRAD1 with a drug, it can actually kill breast tumors grown in mice and in culture. So what we're seeing there is anti-NRAD1 drug applied, but that's just one. We don't know what the other 12,000 are doing and which ones are the most important for breast cancer growth. And then if we figure out which ones are most important, we can best treat those ones. We can develop drugs for those ones specifically and maybe treat breast cancer better. So we've developed a screening strategy which we're applying to figure out which ones are most important. we're going to analyze the expression of all these 12,000 link RNAs in 1,000 breast cancer patient tumors that we have access to for the data of. Then from there, we're going to figure out which ones are most associated with cancer-causing factors, and of those, which ones are most associated with poor survival in breast cancer patients. That will then refine our list of link RNAs that we're going to study in patient samples and see if we can develop drugs that will target them. So now, we've been working on this for the past few months, and next slide. Oh, I'm working on this for the past few months, and luckily enough, the first 3 steps can be done using computers at home, doing computer science, bioinformatics type strategies. So here you're seeing my students MC and Justin working from home during the COVID pandemic. And what they've done so far is they've done their analysis on the first 12,000 link RNAs. They've narrowed it down to 200 link RNAs associated with cancer-causing factors, down to 30 link RNAs associated with poor survival. How do you poor survival in patients is really, really significantly. And now they're thinking, okay, we've got these prioritized link RNAs we're going to study. And so now that we're back in the lab, now, starting this week, they're back. They're there right now. We can test if inhibiting any of these 30 link RNAs are going to kill breast cancer cells. So what we're also doing is in collaboration with surgical oncologists, Dr. Carmen Giacomontoni and Dr. Lucy Hellier, we've been able to obtain patient tumor samples and culture them in cell culture here, and also culture and grow them in mice. And we can then apply now anti-link RNA drugs to the 30 different specific to 30 different link RNAs, and apply them to those patient samples in the culture dishes, and then see what happens. Are they going to still grow, or are we going to stop their growth and kill them by adding these anti-link RNA drugs to the plate that we've prioritized to study these ones? And so we can look with a microscope to see what happens right there and see if we're able to kill these patient tumor samples this way. So the next steps after that, we're going to test. Over there, we're going to prioritize even further and even look even further at the best ones and look at them in animal models. We're going to see if it works better to target these over using currently used drugs. And then we're going to figure out how they do this, how these link RNAs promote breast tumor growth. I'd like to thank you, donors. Thank you, DMRF. You're making this work possible. And thank you to our other funders and my lab, especially doing the work, and then our collaborators as well. So thank you very much. We so appreciate the help.
Hello. Again, thank you, Paula. That was fantastic. I know it was fast. I know it was probably fast for our listeners too. Yeah. We do have a couple of questions. Can you tell us if there are any examples of link RNA based treatments currently in the clinic?
Well, there is actually recently in the past year or so, there's been approval of drugs to be these type of not link RNA specifically, but anti-RNA based drugs to treat neurological type disorders. So we're getting there. So I think that, and then we're also looking we're doing clinical trials now on some of these that are, so I think there's going to be more and more of RNA based targets, and also including link RNAs. We're going to see that coming in the near future.
Great. How far away are we from treating breast cancer with link RNA?
Well, quite unfortunately, we're still at we're right at the grassroots here. We're still just trying to even figure out which ones are the most important for breast cancer. So you can imagine when you start trying to figure out targets, and we don't want to just pick any old one that looks good. We want to test. We want to make sure that we really look at the whole gamut of them, right, to pick the best ones. So I think it's going to be years away, yeah. But we have to start somewhere. It's not going to come tomorrow for sure.
Yeah, no, absolutely. And research that happened 20 years ago or 10 years ago or 5 years ago is having an impact today. So
yeah, exactly.
You're absolutely right. Well, thank you, Paula. I really appreciate all you've given us today. And we do have a question from the audience. Why did you decide to focus your research on the area of breast cancer?
Well, luckily enough, I haven't been personally affected by breast cancer myself. But it was more that when I was working on research, I wanted to, I started working on I was originally working on bacteria, and then I started working on an oncolytic virus. And then I thought, I want to work on breast cancer because to me, I just kept hearing about how many women it impacted. And I just felt that, even though I haven't been personally impacted by it now, I know that it'll impact so many people that I know eventually. And I felt that that would be a way that I could make a difference, that my research could make a difference. And also, it was interesting enough, a lot of the students I interact with, they specifically want to work on breast cancer research when they come to me. So obviously, it's a passionate area for a lot of students. They want to work on this. They wanted to see it. It is a big problem, and so I think that I wanted to make a difference, and that's what I chose.
Thank you, and you are making a difference. We really appreciate you giving us that personal perspective. Question here. You were the recipient of one of the DMRF Breakthrough Breakfast Mid-Career Accelerator Awards from 2018, which I attended. How has this award helped to advance your research?
Okay, so I'd say 3 ways. First of all, what I just told you, that project that I'm working on, it's kind of a risky project, I'd say. So one of the instructions I received when I got that award is go take a risk, take that money and use it on a risky project. And that's what we're doing. So all those link RNA drugs that I'm going to order to test on the patient tumor samples, a lot of that money from the Breakthrough Award is going to be used on that, because the first 3 months that we did, that was all analysis on computers. It didn't really take any money, but this next part is going to take some of that money. The next thing I did is I'm recruiting a postdoc, an international postdoc, which is a little bit of a more risky for me. And so some of that money will also go towards recruiting this new postdoc that's going to come do some research with us. His arrival in Canada has been delayed now. He's not going to be able to come probably till January because of COVID, but that's something else. So recruiting someone else. And then finally, I'm also recruiting another graduate student. So I think as well, so I think that's 3 different ways that I'm applying these funds. But the main drive is what I presented today.
Fantastic. That's wonderful to hear about talent being recruited to the region, and young people really being brought into the fold and being taught research at a young age, and being able to do that here in Halifax. So yeah. Thank you.
And I think for me, and that's why I put those pictures up of my students. It's for me, it's also the next generation, and being able to teach the next generation of scientists science, and they'll take that. So any impact I make will be increased so much more, because we're teaching the next generation, and some of them are becoming so passionate about research, and the things that they'll do all stem from what we're doing here. So it all just grows exponentially. So I feel like I'm happy to be able to do that as well.
Yeah, fantastic. And just in closing, we have one comment here from a donor that says, we all know women who have had to deal with breast cancer. Thank you for the work you're doing in this field. And I think I speak for many of us who have friends and family, or we know someone who's experienced breast cancer. We really appreciate that you're dedicating your life's work to this, and that you're encouraging many other young people to dedicate their life to this. So thank you so much. And we really appreciate you taking the time for us today.
I enjoyed myself. Thank you.
Good. Thank you.
All right. Thanks, everybody. Thanks to the donors.
Okay. Our final Ignite talk features Dr. Rudolf Uher. He's a professor and the Canadian Research Chair in Early Intervention in Dalhousie's Department of Psychiatry. Over the past 7 years, Dr. Uher and colleagues have been leading an innovative long-term study known as FORBOW, and that stands for Families Overcoming Risks and Building Opportunities for Wellbeing. FORBOW is aimed at preventing severe mental illness and supporting positive mental health in youth. Understanding that mental illness such as bipolar disorder, major depression, and schizophrenia have a genetic underline, the FORBOW study has engaged over 500 families in Nova Scotia with a history of severe mental illness to test whether early interventions, either in childhood or adolescence, can interrupt this cycle. Welcome, Rudolf. Now I'll turn it over to you, and the clock starts as soon as your slides come up.
Well, thank you for having me. It's wonderful. Thanks for everyone for turning up. It is a pleasure and a privilege to talk about mental illness here, and for mental illness to get a spot in an event like this it's really great. It's often been forgotten. We have heard about pandemics of infectious disease. We've heard about cardiovascular illness and cancer today. And those two groups of diseases really embody what most people think of in terms of illness and health and medicine. But when it comes to what type of illness causes the most disability and the most lost life years, productive life years, it is actually mental illness, whether we look worldwide or in Canada. Well, the reason why mental illness is the actual pandemic of the 21st century is because of when it strikes, when it affects people. And it's important to look at it in a life course perspective. If you look at this slide, the red line is approximately the growth in human capacity and ability. And it's not a coincidence that the severe forms of mental illness strike exactly when the human capacity is just about to peak, at the end of adolescence and the beginning, or the threshold of adulthood. That's when we see depression, psychosis, and bipolar disorder. These are typically not the first manifestations of ill health. There are precursors in childhood: anxiety, ADHD, temperament. And it doesn't end there. There are consequences later on in life. And when we look at what difference we can make, it's obvious that the earlier we intervene, the bigger return for our investment we can get. So prevention is obviously the way to go, and this is where FORBOW comes in. We presently working with over 500 children and youth and their parents. The sample has been collected in Nova Scotia over the last 7 years and greatly supported by DMRF and many of the donors who I'm sure some of them are on today. And FORBOW aims to reduce the risk of mental illness and the burden of mental illness in two ways. We first want to identify risk early. What it takes is what we know: family history. Mental illness runs in families, and that's why we're working with families. And there are early signs, those precursors, antecedents that we can identify in children and adolescents. There's genes, of course. The family risk is substantial, from 10% risk in the general population to 30% or 1 in 3 if one parent is affected, and 50% or 1 in 2 if both parents are affected. The other way we can get at the gene is to look at that risk is to look at genes. And we use saliva samples. This is a saliva collection tube to measure genetic variants that contribute to the risk of mental illness. And they do predict who is likely to develop mental illness. And these people are not doomed to develop the disorder. It's a probability that we need to work with in terms of prevention and skills that will help them stay healthy. The genes are reflected in brain development, and we've been fortunate to use the great new facility here in the QE2 hospital and the Biotic Center to measure the development of brains over time. One thing we can do with the brain scans that's fairly simple is to estimate how old the person is with a precision between 1 and 2 years. Now, you can see most of the bubbles are close to the line, but some of them depart. And those people who have brains looking older or younger than their actual age are at high risk for mental illness. We're looking at the analysis of speech. The human thought comes out in speech, and human brain shows how it works in speech. And we work with our colleagues in Dalhousie Computer Science to analyze speech in a way that can detect the risk for mental illness early on and shows us who needs intervention and who we should work with. In this case, we can see risk for bipolar disorder by greater difference in sadness in speech in response to positive and negative prompts. Well, when we pull all this together, the genes, family history, early signs, brain, speech, we get a pretty good prediction in terms of who is likely to develop mental illness. But it's only worth identifying risk if we can intervene and change it. And that's exactly what we're doing in FORBOW. We are currently testing a series of interventions for different ages, starting in preschool years, focusing a lot on adolescents, but also introducing early personalized treatment for those who develop mental illness in their late teens or early 20s. Skills for Wellness, or SWELL, is a skill-enhancing psychological intervention for youth starting at age 9 and age 9 to 19. And as we saw, it's reducing symptoms. Coach, Courageous Parents, Courageous Children, is aiming even earlier. Here we're working with parents of children in the preschool age to stop family transmission of anxiety that is one of the antecedents that leads later to mood problems and psychosis. Break it early. And with any one of these interventions working, well, we need years to see the results. It's a long-term work. It may take 8 years to find out what's the effect of the interventions that we're delivering now. But it is our estimate that up to 7 out of 10 cases of mental illness are avoidable. Thank you.
Thank you, Rudolf. I know that's rapid pace, and I know you have lots to say about this amazing project. We're a little short on time, but I really have a couple questions I want to get in. Many of our donors have supported this project.
Yes.
Tell us how a single donation can make the greatest difference for preventing mental illness.
Well, preventing mental illness takes years, and no research grant lasts 8 or 10 years. And that's what we need to see a young person through their risk periods, through the adolescence, and keep them healthy. And the great advantage of DMRF was to providing the continuity, knowing that we can work not just for the next year or two, but that we can plan in the life course pattern.
Great. Another question we have is actually from one of our donors. I'd be interested to know one of the key positive breakthroughs thus far that has really pleased your team and helped you to propel forward and keep you motivated in your work.
One of them has been the uptake. When we proposed this work for the first time, our fellow researchers said, this is ambitious. And that often means, it will not happen. And one of the concern was that we're going to be offering interventions to people who are not yet ill and not yet treatment seeking. And say, well, if you do that, not many of them will do it. Well, the uptake of this intervention is well over 80%. So the families that we are working with, they trust us and they want what we're offering them. And they do it because they see benefits in real time, in the short term, not just in the long term. So seeing this work and knowing that it will be possible to find out how much we can reduce the long-term risk of serious mental illness, that really has been a major breakthrough that keeps us going.
Fantastic. Thank you so much, Rudolf, for today. I know that was rapid fire, and we really do appreciate your groundbreaking research. We appreciate your team, and we're so pleased that we can support this work and that our donors can. So thank you.
Thank you, everyone.
So we have a comment from Christine Chambers, who actually was one of our Ignite speakers last year. We put her through the rapid fire questions, and she said that she loves this virtual impact reception. Thank you for hosting this. And now we're going to move on to someone else actually who was here last year. Dr. Craig McCormick was also one of our Ignite speakers. We're really excited to present a bit of a Q&A. Craig is not going to give another Ignite talk today, but Craig is a Dalhousie professor and a virologist. And if you follow DMRF, you know that Craig has actively participated in many of our fundraising campaigns. He was also in the hot seat last year, as I said, and he did quite well. And luckily, he's a fast talker like me. But while Craig's work has focused largely on the study of two viruses, the influenza virus and a cancer-causing herpes virus known as KSHV, when the COVID-19 pandemic hit, he also added research in this disease area to his portfolio. Craig was also one of the 40 researchers whose work was supported by the Nova Scotia COVID-19 Health Research Coalition. And we'd like to ask him about that today. So Craig, thank you for joining us.
Yeah, thanks very much for having me. This is a nice opportunity.
Great. So we have a couple of questions, and if any come in from the audience, we'll try and squeeze them in. Craig, we want to thank you first for the awesome video you did that many of our donors would have seen with your daughter, Sophie. Sophie, who's also a donor to DMRF herself, and we're very proud of that. She's 14?
She's about to turn 13.
13, great. And you created that video to help us kick off our COVID-19 series. And we really wanted the kids in our community to be able to hear from one of their peers and from a researcher about this, scary pandemic that they've been hearing about, and it disrupted their lives. So we really appreciate you doing that. What was it like to appear in a viral video about research with your daughter? What was the reaction from your family and friends?
We got really positive reactions from family and friends, and actually it's continued. So I think it's been a couple of months now, but we still, as things open up, we get more and more positive feedback from people we hadn't seen in a while. A lot of them tell us that they really liked the level that we pitched it at. So obviously Sophie is she's not little. She's 12. She's almost 13, and so she understands a lot. And so we tried to have a conversation that was targeted towards somebody like that, who has some understanding of the world. So, we heard from children, but also adults, that everybody learned something. So I think that I was happy with how that turned out.
Yeah, fantastic. Thank you for doing that. And please thank Sophie for us for both doing that and being a donor. We really, it's fantastic to see young philanthropists learning at that age that giving back is important, and investing in research is, going to benefit her in, 20 years. So I really appreciate her.
We'd be very happy to do it again. And she's told me already that if we do a follow-up video, she'd like a little more creative control.
Fantastic. We can make that happen. As mentioned earlier, DMRF was proud to be one of the leads in the creation of the Nova Scotia COVID-19 Health Research Coalition. Craig, can you tell us a little bit about the project that you're working on as it relates to COVID-19, and what some of the proposed impacts might be from that project?
Right. So first off, we're just really grateful for the support of the DMRF donors who've made this research possible and have supported it this year. It's been a really busy time in the lab, and I'm very proud of my team and what they've accomplished so far. As you know, so we're molecular virologists. So what that means is that we study viral genes, and we try to figure out how they work, and how they give the virus an advantage in our bodies, and maybe help the virus hide out from the immune system, that sort of thing. And so when the pandemic hit, we pretty quickly acquired all of the viral genes for SARS-CoV-2 so that we could do the kind of things that we do, like to figure out how different genes contribute to viral replication, contribute to the disease process. So sort of a reductionist approach, but really important. So coronaviruses are sort of medium-sized viruses. They've got about 30 genes. And you can imagine that, for only 30 genes to have such a profound combined effect and cause such a devastating disease, some of them are very important. So we're still at the early stages of figuring out how these different viral genes work in order to help the virus, again, hide from our immune system, cause, elicit inappropriate inflammation that might be tied to the disease process. So yeah, we're trying to understand those disease processes. In addition, I just wanted to say, one side benefit of starting to work with these viruses and their genes has been our ability to contribute to some of the other work that's going on sort of collaboratively in the team. So earlier you mentioned the vaccine development with Dr. Roy Duncan and Dr. Alison Kelvin of the DNA-based vaccine that they're developing for COVID. Some of our manipulating and producing viral genes and viral proteins has been useful for allowing us to contribute to that vaccine development work.
Well, here's the old switcheroo, Craig. Joanne froze, so we've stuck me on the camera. So it's a good thing I'm wearing my DMRF branded shirt today, I guess, right? And have a lovely DMRF branding behind me. Hi, everybody. I'm John Broom from DMRF. I'm Manager of Donor Relations. While Joanne's trying to get back on, I've got one last question for Craig. So, Craig, our impact report touches on the DMRF Mali appeal and how its many impacts over the last few years. So donors to the 2018 campaign helped to fund the purchase of the ImageStream X Mark II. What can you tell us about this piece of equipment and how it's helping your research?
Yeah, so the ImageStream is, we're again grateful for the support because, one thing about Dalhousie is we have access to really outstanding equipment, scientific equipment that that has, is well run, well supported, and the ImageStream is just it's the next latest addition to a very impressive suite of equipment. And so basically what it is, it's a sort of a powerful hybrid instrument that combines the ability of microscopes to peer deeply into a cell and tell you something about what's going on in that cell, whether it's a cancer cell or a cell that's being infected with a virus. So combining the power of advanced microscopy with single cell analysis. And what I mean is the ability to look quickly and characterize individual cells and this is a process called flow cytometry. So what this instrument does is basically marries those two technologies together so that you can get a peek at the interior of a cell amongst a population of cells and learn something about its properties. For example, if you had 1 million cells in a container and maybe only a handful of them were doing something interesting that was helping them fight off a virus, this technology would allow you to identify those special cells that had some sort of advantage over the virus. And then, so being able to find that needle in the haystack allows you to characterize what's going on in those special cells. And then maybe there's some generalizable information that we could get from that we could use to fight off a virus better or understand the workings of a cancer cell. So the technology is in place, and we're excited to use it actually to support our COVID research.
Awesome. Thank you so much, Craig. We have one question from the audience, and I'm going to pop that up. While we do that, we're going to tag me out and tag Joanne back in, but I'm going to pop the question up for you right now. It's from your colleague, John Archibald. His question is, what is the current scientific understanding of where COVID-19 virus comes from in nature?
Yeah, so surprisingly, we don't yet know. There has been a lot of investigations of this. There's obviously a lot of interest in which animal reservoir in nature the virus might have come from, whether it went through an intermediate host to get to us, and so basically there's been a few false starts. There have been some viruses that are similar but not identical that have been identified in a number of different animal hosts, and I think that we just don't know yet. And we're going to get there only through a lot of environmental sampling in Southeast Asia, and that will help us to identify, hopefully over time, the animal reservoir. So there have been hints. We've learned a lot about relationships between these viruses, and I think that with time we'll get there in terms of figuring out the origin. What it speaks to is that we need lots more funding for research of this kind, and as you know, some of these larger projects in surveillance were terminated this past year just before the pandemic hit, and so the funding was withdrawn in exactly the wrong time. We need to keep supporting this kind of work.
Yeah, great. Thank you so much. Sorry you lost me there for a second. Not sure what happened, but I'm back in the game. And I could hear you the whole time, so that's great. Thank you, Craig, for joining us. That was very meaningful, and we certainly appreciate that work that you're doing on a number of fronts, influenza, cancer, and on COVID-19. So thank you for being here today. Say hi to Sophie for us.
Very good. Thank you.
Thank you. And now we're going to have some prize giveaways. Our prizes aren't new cars, unfortunately, but we do have a DMRF journal, an iPack so that you can get lots of sleep, and a travel toothbrush. And so those 3 prize packs will go to John Archibald, Gloria Shabel, I think is how you pronounce it, and Greg Sutton. And then we also have our main prize giveaway is a DMRF windbreaker that you can wear golfing or anywhere on a vacation. Of course, the nights in Nova Scotia aren't too warm, and that one goes to Judith Case. So thank you so much for commenting and being engaged and asking questions here today. So now we're going to pass the floor over to our just past chair of our board, Alan Shaw. Alan is a consummate business person who has been a member of our board for 16 years, 4 of them as our board chair. Alan and wife Leslie are also generous donors to DMRF who believe in the value of health research and have supported melanoma research over the last number of years very generously. Alan's business acumen, wise counsel, and dedication to advancing local health research has been crucial to our organization's ever-growing successes. In June, Alan ended his tenure as chair of the DMRF Board of Directors, allowing him more time with his children, grandchildren, and his lovely wife, Leslie. Luckily, we will continue to benefit from his leadership as he's staying on our board for a while longer. I know I've really enjoyed Alan's time as chair. He's taught us all so much and really truly believes in our core mission of improving health outcomes through health research. Alan, thank you so much for all of your support, and I'll turn it over to you to say a few words here today.
Unfortunately, we seem to be having another technical difficulty there, Joanne. Surprise. Alan, we don't seem to have him at the moment. So sorry, Alan. Maybe, Joanne, you could introduce our new chair.
Absolutely. I will introduce Janet McMillan. So Janet McMillan is here today, and Janet has been on our board for a number of years. I've appreciated working alongside her for the last 4 years. Janet, you're an amazing professional, and I'm certain you and I are going to have a lot of fun during your time as chair, and you've come in at a fantastic time, and I think it's going to be great. So if you want to say a few words, and if Alan comes on, we'll bring him in.
Great. Well, thank you. Thanks so much. I can't tell you how much I've enjoyed serving on the board of DMRF since I joined in 2013, and I'm really honored now to serve as the foundation's newest board chair. Every year, I am blown away by the dedication and the passion of our researchers and staff and volunteers. And I'd like to thank you all sincerely for your time and efforts. I've come to learn that there's no greater impact on our health than investing in research, and it crosses all the spectrums from viruses, cancer, mental health, and heart disease. You name the illness, the disease, or the challenge, health research has the ability to touch all those areas, and we heard about that today from some of those excellent speakers. Each and every one of us has been impacted by health issues, whether it's a loved one, a colleague, or ourselves individually. And we know that health is crucial to our survival as a species. So this has been even more evident. This has never been more evident, I mean, than in the past 6 months, as we've watched and lived the dramatic effects of the global health pandemic. So while average people like you and me can isolate and social distance and put on our masks and take as many precautions as possible, it's the health researchers who will end this crisis. It is the health researchers who are going to find long-term solutions to the complex and devastating impacts of the novel coronavirus and the disease COVID-19. So I'm so grateful, and we are all, I think, so grateful. The excellence of our local health researchers has never been more evident than when recently Prime Minister Justin Trudeau announced mid-May that the first Canadian clinical trials for a potential COVID-19 vaccine would be led by researchers at Dalhousie University, and we should all feel great pride. Many of these researchers you've already supported through your gifts over the years to DMRF, and today I want to thank all the researchers for your painstaking work. We appreciate you more than we can say. And to you, our donors, just wow, your giving is propelling this work forward. You are investing in your future and that of those that you care about. And that's probably one of the main reasons that I'm so honored to be serving as DMRF's volunteer board chair. I look forward to big things ahead in the coming years. So on that note, I'll pass the floor back over to Joanne, and hopefully we might have Alan in our midst. Joanne, I'm excited to be working with you over the next several years. I've loved our past few years together. I enjoy your passion and experience, your expertise, your vision, your tenacity. All those things make you an exemplary choice for leading this organization into the future.
Hello, I'm Alan Shaw, the guy that's been missing. Thank you, Joanne, for those very kind words, and thank everyone for joining us today. I am proud to be a long-standing member of the DMRF board, as well as board chair for the last 4 years. During this time, our foundation has grown to support more areas of research. We've also expanded our internal resources to be better equipped to raise more funds, to pay out more dollars to researchers whose mission is to find faster diagnosis, better treatments, and ultimately cures for the many conditions that impact all of us. For those of you who might not know DMRF's history, over 40 years ago, a group of community leaders came together because they saw the immense potential of Dalhousie University medical researchers to advance their outstanding and life-changing work. They just needed the right support. With visions of keeping outstanding health research vigorous and local, our foundation was formed. Today, we have raised tens of millions of dollars to support local health research, and that is thanks to your continued financial commitment. I repeat that again. None of this would be possible without your continued commitment financially. Thank you so much. I will continue to be a member of the DMRF board because I believe in the power of health research to impact our health and our wellness. We all know that good health is the foundation for a good life. That's why my wife Leslie and I will continue to be DMRF supporters and donors. Now it's my pleasure to introduce DMRF's new board chair, Janet. I've appreciated you for the last 4 years. Your intelligence, compassionate nature, strong vision, and outstanding communication skills have and will continue to serve the foundation well. Professionally, Janet serves as lead strategic counsel for National Public Relations and is an expert in strategic planning and governance, stakeholder engagement, corporate communications, and crisis and issues management. Janet, welcome, and thank you for taking on this challenging and most rewarding role. Over to you, Janet.
I'm actually going to take it over from here, and Janet's already had a few words, and I so appreciate both of you, Alan and Janet, for your time and your commitment as volunteers to this organization. You certainly go above and beyond to forward our mission and to help us succeed. So thank you to all of you for participating today. To our researchers, I know we put you through the paces with really fast fire action presentations. If anyone wants to learn more about their research, if you have questions, if you want to hear from the researchers, please reach out. I know it was just a bit of a taste of their work, but we're more than happy to provide you some more information about what they do. I also want to thank my colleagues at DMRF who commit so much of their time and energy to this social purpose and to our social profit, and want to thank them for bringing this impact reception to fruition today. Mostly, I'd like to thank you, our donors, and our viewers for joining us. I hope you've enjoyed this sampling of health research that's taking place because of you, and that you understand just how grateful we are for your support. You're truly having an impact on the health of everyone in the Maritimes and beyond. On that note, don't forget to read the impact report. You can find that at dmrf.ca/impact. And now to play us off, I know you all enjoyed Nevin. Classical guitarist Nevin Prostrand is here to play for us a little while longer. Thank you, Nevin.